All right. All right, so this is topic 7, right? Cell cycle 1, and this is going to be session 2 of that. So as I stated on Thursday, for cell cycle, we have 2 units that we're going to be going through, right? So topic 7 this week was regulatory mechanisms within G1. And next week, we're going to be talking about regulatory mechanisms as well as important cell checkpoints in S, G2, and M phase, right? So remember, make sure you have a good understanding of this week's material because it's crucial for understanding next week's. It's like absolutely crucial. So just make sure you really understand this week well. And like I said before, I have this reminder here, notice how a lot of these regulatory mechanisms work around DNA and make assumptions to why that's important. So on Tuesday, the slides were more content. So if you want, I do have content in these slides, but if you want more content and going in more detail, watch the Tuesday video on YouTube. This one's going to be content, but try to add a lot more questions in. All right, so I showed you this cell cycle overview slide, right? And on the right-hand side, we have a bunch of just little tidbits of information about what's happening in each of the phases of the cell cycle, right? So usually, a lot of our cells right now are chilling in G0, right? They're not dividing. They're just surviving, right? If you will, right? So when a cell receives a growth factor signal, it's going to enter into G1. We call G1 our first gap phase, our first growth phase. The cell's going to grow. It's going to check for DNA damage. And then we're going to run into, our end phase checkpoint, right? Which is called the restriction point or start in yeast. So after we pass the R point, we're going to be in G1 for a little bit longer. And then we're going to pass into S phase, right? And S phase is when we have DNA replication. We also have our end phase checkpoint. After S phase, we're going to go into G2, which is our second growth phase, right? And the cell is going to prepare for chromosome segregation. And we also have our end phase checkpoint in G2. And then our final phase to wrap everything up is M phase, right? So that's where we see PMAT, right? The prophase, metaphase, anaphase, and telophase, right? And we also run into an important checkpoint, the spindle assembly checkpoint. So I have a little note here that I added. So if a cell does not pass through the restriction point, it can re-enter G0 and start G1 again when ready. So if the cell is, in G0, let's say it gets a growth factor signal, it's in G1, but let's say, we don't have enough nutrients to do the cell cycle. The cell can re-enter back into G0. But if the cell already passed the restriction point, that's when apoptosis is going to happen if the cell cannot continue to go through the cell cycle, right? So that's just a piece of information that I added based on a couple questions. All right, so here is our first couple questions, right? And these beginning ones are a little bit, I think, on the easier side, but it's good to make sure you know these ones. So as before, I'll just read it and then I'll answer it. And then if you have any questions about it, just interrupt or let me know, all right? So the first question here is, in which stage of the cell cycle, according to Bio 285, is DNA being replicated, right? And remember, this is going to be in S phase. So it can be a fuzzy subject depending on who you ask. But for this class, Laura defines DNA replication in S phase. So our second question here is, in which phase are the cells considered to be in when they're not replicating, right? Either permanently or temporarily. So we're going to define this in this class as G0, right? That's what a lot of your cells are doing right now. Not all your cells aren't always dividing all the time, right? They have a latent period where they're just surviving, right? So question number 2. So we have 3 loosely defined stages in G1, right? We can do it as early, mid, and late. So I want you to apply these to each of the following questions. Oh, yeah, David, you have a quick question on 1?
Yeah, I do. So 1B specifically, it asks when the cells are not replicating, right? So I know that G naught is when, or G zero rather, is when like the cell has not passed the restriction point. So like it's not, entering that cycle. But is there any replication going on in G1 and G2? Or is it just like that growth and those checks for continuation?
So maybe I actually pulled this question for Laura. I guess maybe the wording could be a little better. I guess if think about it as like, in which phase here is the cell not actively within the cell cycle to create two daughter cells, right? So
Okay.
I guess that's what I kind of meant by replicating. I can like reword that before I send the slides out, but yeah,
Okay. Yeah. No worries. All right.
Yeah.
Thank you.
no problem. All right. So like I said, 3 loosely defined stages apply to each of the questions, right? So in which part or parts of G1 is cell division signals received through the RTK RAS-MAPK pathway, right? So we would define this as an early G1, right? So that that growth hormone signal is going to be perceived by this pathway, and that growth hormone signal is going to result in us getting our first production of cyclins, right? So the second question I have here is, in which parts of G1 is the cell growing? So this is, I guess, can be like a little bit of a trick question, but it would be the entire G1 cycle, right? Early, mid, and late. The cell's constantly growing throughout G1, so it's no specific phase. My third question here is, in which parts of G1 is the cell checking for DNA damage? So another important one, like I said, these were previously exam questions, so I'm going to put them on these slides, but this is early, mid, and late, right? So, but a side note here is that cells specifically check for DNA damage in early to mid G1, but on an exam, you would never be docked points if you said early, mid, and late, or the entire G1 phase. All right, question D. In which part of G1 is CDK active, right? So this would be late, right? So remember, CDK activation allows the cells to enter late G1 after the restriction point. And a side note here, remember, it's important that CDK is not active in early to mid G1. Why, might you ask? Because CDK inhibits the formation of the pre-RC throughout the rest of the cell cycle. This will make a lot more sense here, D, next week, because next week, Laura is going to teach you the mechanism of how active CDK cyclin complex inhibits the reformation of the pre-RC. So if you still don't get why D is the answer, just know that next week it will definitely come to you. All right, and last question here. True or false? After the cell passes the restriction point, R or start in yeast, the cell has entered G1, right? So, oops, false, right? Remember, the cell is still in G1 after the restriction point. This is kind of just like a fact, right? All right, any questions on any of the ones on question 2? All right, question number 3. So in which phases of the cell cycle do the following events occur? And we have G1, S, G2, and M phase. So in which phases is spindle forming, right? So this is going to be M phase. A lot of these questions are kind of just like factual based. I really don't have like an explanation of why. It's kind of just like, the fact that spindles are forming in M phase, right? So in which phases is the cell growing? So this one would be our G1 and G2 phase, right? These are our gap phases. These are our growth phases. In which phases is the spindle assembly checkpoint, right? Spindle assembly checkpoint. I just told you that spindles form an M phase, so we can deduce that the checkpoint's actually also going to be an M phase. D, in which phases do checkpoints exist, right? So remember, the cell cycle has to be highly regulated. So we would expect checkpoints in every single phase of the cell cycle, right? G1, we learned about the restriction point, and next week we'll learn about checkpoints in S, G2, and M phase. So our final question here is, which phase is considered the beginning of the cell cycle? So like I said, we'll define the beginning of the cell cycle as G1 phase, right? Our first growth phase, and our first gap phase. Is there any questions on question number 3. No. All right. Question number 4, short answer. So this is actually, I believe this is a problem set question. So make sure you double check your answer on your problem set if it doesn't align. So question number 4 states that RNR1 is a gene that makes ribonucleotide reductase protein, which is required to synthesize nucleotides for DNA replication. Hydroxyurea is a molecule that inhibits the function of this enzyme. If you were to add hydroxyurea to your cells, in which phase of the cell cycle do you think the cells pause? So whenever you're doing a question on the exam, this is what I always do, right? So what information should I pull from the question, right? I think the first important piece of information is that RNR1 is a gene that makes the protein needed to synthesize nucleotides for DNA replication, right? I took a lot of the junk out of this question, right? We don't need, to answer this question, we don't really need these, fancy words, ribonucleotide reductase protein, right? We just need the bare bone information from it. And the second important information is hydroxyurea is a molecule that inhibits the function of RNR1. So if you were to add hydroxyurea to your cells, in which phase of the cell cycle do you think the cells would pause, right? So let's see. So when HU is added to cells, they'll halt their cell cycle in S phase, right? So this is because without sufficient nucleotides, DNA polymerase cannot duplicate DNA, right? So that's the main question. Now, the second bullet point, or that's the main answer, the second bullet point here kind of gives someone talked about in class, like, well, why wouldn't it be G1? And to be honest with you, it's kind of just, it just doesn't exist, right? So as you see here, as a side note, RNR1 is typically upregulated in G1, but there is no checking mechanism for sufficient nucleotides in G1. So the cell will progress into S phase and will have to pause there. So G1 would be a very good hypothesis, but it's just a fact that there's no checking mechanism in G1. So any questions here on 4? All right. So those were, I guess, the main questions relating to just the phases of the cell cycle. So now we're going to get into the cell cycle control system, right? So I'm just going to run through some slides that I did on Tuesday, just for maybe like a little bit of a refresher. So remember, the cell cycle control system is regulated by cyclically activating and deactivating proteins and protein complexes required for initiation or regulation of DNA replication, chromosome segregation, and cytokinesis. Remember, cyclically, a circle, it's on and off, on and off, on and off, right? And this is done by two categories of enzymes, right? We have kinases and we have phosphatases. Kinases add phosphate groups, phosphatases remove phosphate groups. And a key point, like I said, phosphorylating doesn't always mean activating. So make sure you remember that, because there could be an exam question that might say, like true or false, phosphorylating always means activating a protein. So make sure you know that that's not true. So our main kinase that we see in the cell cycle is our CDK, right? Our cyclin-dependent kinase. So CDK, we call it the master cell regulator, right? So it's a serine-threonine kinase. And as I said before, serine-threonine kinase means that CDK, when bound to cyclin, is going to bind ATP, and it's going to cut off that third phosphate group, and it's going to add it to serine and threonine hydroxyl groups on target proteins, right? So it basically adds phosphate to serine and threonine amino acids. That's basically what CDK does. So CDK can be regulated in a couple different ways, right? It can be regulated with cyclin levels. It can be regulated through phosphorylation, through like CDK activating kinase or WEE1. It can be regulated through CDK inhibitors, right? And we usually see these in G1, where we see them responsive DNA damage, right? We learned about P53, which is a CDK, which codes for, it's a transcription factor that codes for a CDK inhibitor, right? And we also have dephosphorylation, right, by phosphatases. So to activate CDK, we have two events that need to happen, right? We need to bind cyclin, and CDK needs to be phosphorylated by CDK activating kinase, right? And remember, if the cell's ever not going through the cell cycle, or the cell's ever pausing in a specific phase of the cell cycle, that means that CDK is either being deactivated, like maybe WEE1 phosphorylated it, or a CDK inhibitor is bound to it, right? Without these mechanisms, the cell would constantly go into the cell cycle and there'd be no regulation. So let's go a little bit more into detail with cyclin, right? So remember, cyclin is required for CDK kinase activity, right? So cyclin activates CDK kinase activity. It also tells CDK what to phosphorylate, right? So it gives that binding specificity, right? So both cyclin and CDK are going to make an interaction on the target protein. So cyclin regulation. So cyclin is regulated on the transcription and translational control, right? So if we need cyclins for, S phase, we're going to transcribe and translate a bunch of cyclins, right? And then when we don't need those cyclins for, G2 phase, we're going to use a ubiquitination, right? Ubiquitination, tag cyclin, goes to the proteasome, gets degraded. Sorry, guys, I'm just drinking some water. All right. So we also learned about ways that, or not ways, but we have this mechanism that we learned about how cyclin CDK become active, right? So this has to do with the T loop, right? So we have inactive CDK. It's going to bind cyclin. Cyclin's going to pull back this T loop, which is, you could say something along the lines of this T loop could be a domain, right? Inside CDK, right? So cyclin's going to pull back this T loop. It's going to reveal an ATP binding site, right? So now ATP can diffuse in, and this complex can go and phosphorylate downstream proteins. And then CDK activating kinase can also phosphorylate the T loop to stabilize this conformation. All right, so now let's talk about these two as a complex, right? So CDK functions throughout the cell cycle with different cyclic partners. So for the most part, CDK levels stay the same throughout the cell cycle, right? So, in G1, S, G2, M, CDK levels are going to roughly be the exact same. It's the cyclin levels that are going to change. And that's what gives us the specificity, right? That's what allows this point right here, right? How to get the same CDK functioning throughout the cell cycle and targeting different proteins in each phase, right? That's cyclin. So now that we know that, right? First of all, any questions on any of that before I move on? I know I went through a little fast because I did it on Tuesday, but any questions on any of that? All right. So knowing that, let's get into some questions, right? So is the following an example of a way in which the cell regulates the cell cycle? Yes or no, and explain. CDK is cyclically phosphorylated and dephosphorylated by other kinases and phosphatases. So yes, right? So CDK is regulated by phosphorylation and dephosphorylation. We know that one way to regulate it through phosphorylation is through CAK, right? CDK activating kinase. And one way to regulate it through dephosphorylation is with, or not dephosphorylation. Another way to regulate it through phosphorylation is WEE1. And another way to regulate it through dephosphorylation is CDC25, right? So CDC25 actually removes that phosphate group put on by WEE1. Any questions on 5? All right, question 6. So this is another, I think, like a question you guys had in class. And this actually came up in my exam when I took this class a couple of years ago, which is cyclin is considered necessary for CDK, but not sufficient, right? So for CDK to become active, it needs to bind cyclin and be phosphorylated by CAK, right? So what does that mean, right? So cyclin is necessary for CDK to be active, but it's not sufficient enough. If CAK doesn't phosphorylate this complex, we don't have an active CDK. So it's necessary, but it's not sufficient, right? Any questions on 6? All right. So I'm going to quickly run through this cell cycle arrest system, right? So this pathway with P53 as a transcription factor is an example of a CDK inhibitor, right? So we're inhibiting CDK cycling complex from moving through the cell cycle. So this pathway kind of starts off, as you see here, it says your cell finds a DNA mutation in G1, right? So X-ray, UV light, sunburn, you have DNA damage, right? So when DNA damage happens, a kinase cascade, which I don't know what kinase cascade it is, but a kinase cascade is going to add a phosphate group onto P53, right? Remember, P53 is a transcription factor. And when this phosphate group is added to P53, it becomes stable and it becomes activated. So since P53 is a transcription factor, it binds to the promoter of a gene called P21. And when it binds to the promoter of the gene P21, it's going to recruit RNA polymerase, which is going to make our RNA transcript, which then ribosome is going to bind and make our actual P21 CDK inhibitor protein. So, knowing that, we have a question, right? So I think this is also might be a problem set question. Oh, I see something in the chat.
Yeah, sorry, I just have another question. So you said that P53 binds at the promoter region
Yes.
and recruits RNA polymerase, correct?
Yes.
Okay, awesome. Thank you.
Yeah, so it's a transcription factor. So that's kind of conserved through transcription factors, but yeah, that's what the main idea of it is. So
Awesome. Thank you.
we have this question here, which I'm going to just sip water before I read it because my mouth is getting dry. All right, so P53 is constitutively degraded when not active. Constitutively means continuous. I know some people on exam 1 asked a lot of questions about, like, I think it was even constitutively was actually one of the words that people were asking questions on. So remember, on an exam, if you ever don't understand a word, make sure you ask, because this word, knowing this word is very important for understanding the question, right? So what would be the result if P53 was degraded even when active? So say a gain of function mutation, ubiquitination ligase that targets P53. Would this cause an increase or decrease response to DNA damage, and would this lead to a cancerous phenotype or to programmed cell death? So this would lead to a decreased response in DNA damage, right? Because P53 isn't available to respond. And this is essentially a loss of function mutation, P53. And a decreased ability to respond to DNA damage is something that can lead to a hypermutation phenotype, which we can contribute to a cancerous phenotype, right? So is there any questions on question number 7? All right, so now we have some mechanisms of control and regulation in G1. So I gave you this list on Tuesday. Very important, because I'll tell you right now, where Laura is going to test you really hard is if you know when things are occurring. Now, obviously, some stuff in this is overlapping, right? The cell's constantly checking for DNA damage. I just happen to put in the number 3 position, right? But the important ones to understand is, like when that the pre-RC forms before activation of CDK, right? And the activation of CDK is needed for, the restriction point, and that, the pre-RC needs to come after CDK activation. So that stuff, some stuff overlaps, but in general, I think this is a good study aid. So I definitely would make sure, I don't know if Laura even still gives you guys a cheat sheet. I know like maybe a couple semesters she has, but I don't know if she's still doing it or not. But if you do have a cheat sheet, good piece of information to put on it. So the first thing I'm going to get into is the pre-RC forms in early G1 phase, right? So when cyclin CDK levels are low. So Iris went over this on Tuesday. I'll just run through it really quickly. We have our DNA, right? ORC is going to bind to it. ORC's an ATPase, right? And once ORC binds, it's going to recruit CDC6. right? CDC6 is going to recruit the MCM complex with help from CDC1. And then once MCM is binded to CDC6, which is bound to ORC, CDT1 is going to dissociate, right? And just remember that ORC is ATPase, MCM is a helicase, right? So helicase is an enzyme that takes two DNA strands and separates them for DNA replication, right? So once we have this pre-RC formation, this is called origin licensing, right? Any questions on this mechanism here? All right. So the cells after this is, those are this up here, but I want to talk about a little bit more about E2F and RB, right? So another little mechanism that we learned, right? So active CDK cyclic complex is going to come and phosphorylate RB, which is going to let go of E2F, and then E2F is going to transcribe and eventually help translate or help transcribe, and eventually they're going to become translated, right? And these are going to be S-phase proteins, right? So RB gets phosphorylated by activated cyclin CDK, E2F is released and induces the transcription of these S-phase proteins, right? And remember, this is the restriction point. This is the point of no return. Any questions on this pathway?
I just have another one real quick. So what is like the function of the pre-RC?
So it's, think of it, have you heard of like the origin of replication?
Only in the context of like the last slide, to be honest.
Yeah, so the origin of replication is, you're going to learn a lot about this in like 2 weeks, I mean like 2 or 3 weeks. But the origin replication is just a bunch of proteins that are needed to replicate DNA. So this pre-RC is kind of like our first half of the proteins we need for replication. And then when the pre-IC forms, which I'll get into in like a couple slides, that's like the rest of the proteins, so we can actually start replicating DNA. Does that make sense?
Yeah, so does E2F have anything to do with the pre-RC, or is it just the proteins that it eventually induces transcription of that go to the like pre-IC?
So I've asked Laura this. She has to check in guess some literature, but from my knowledge, I think the proteins that are made, the proteins that E2F induces the transcription of, I believe those are the proteins that are added onto the pre-RC to form the pre-IC.
Okay, all right.
So, which would make sense, right? Because the pre-IC.
Right.
I'm not 100% sure, and she's not 100% sure, but I mean, for me, I think it makes sense. So.
Yeah, I agree.
But I'll ask her one more time for that. I know she for it on Tuesday. But
all right, thank you.
No problem. Anyone else have any other questions on this pathway? All right. So, in our final step here, right, was that proteins were added to the pre-RC, right? So, another thing just to put here, don't memorize the proteins for the pre-IC. You'll never get asked them. Do know the proteins for the pre-RC, right? Do know, ORC. Make sure you know ORC. Make sure you know CDC6. Make sure you know CDT1. And make sure you know MCM. They're going to come up next week in S phase, right? But make sure you know these proteins. But do not memorize the proteins for the pre-IC. I don't know any of them either. So don't memorize any of these proteins. Don't put it on your cheat sheet either, because I think it wastes space as well. Right. So proteins are going to get added to the pre-RC, and we're going to get the pre-IC. Right. So that's kind of the step right before we start DNA replication. So knowing that sequence of events, right, we have question 8, right? So which of the following are examples of way in which the cell regulates the cell cycle? Yes or no, and explain. So CDK phosphorylates RB, which causes it to bind E2F, which leads to transcription of S-phase genes. So, as I said, no, right? So CDK does phosphorylate RB, but that causes RB to dissociate away from E2F, right? So, and which causes E2F to be active and upregulate transcription of S-phase genes, right? Any questions on A? All right, B here. So pre-replicative complex, right? The pre-RC forms of the origin replication during S phase. No. The pre-RC forms on origins of replication in G1 phase, and they are inhibited from reforming on origins in S phase. So make sure you understand that. And like I said earlier, it'll make more sense next week. Any questions on any of those? All right. Question 9. Are pre-RCs, this is actually, Laura asked you this question in class today. So are pre-RCs formed before or after the restriction point? So this would be after, right? Remember, because we need active CDK cyclin complex, which happens right after the restriction point to form the pre-IC. So does anyone have any questions on 9? No. All right. So I kind of just have a diagram here. So, we form the pre-RC for the pre-RC first, then CDK has to be activated in order to form the pre-RC, right? Because active CDK recruits these proteins for the pre-RC. All right, so that's kind of just going over it. And I don't know, someone said, I don't know if Laura talked about WEE1 in CDC 25 this week. That might be a concept that she might talk more about next week. But I think we just add it to the slides, because I think it just makes more sense for understanding the material. All right, so I think this is the last prepared question I have, right? Question 10. So question 10, A through D, yes or no, would RB being phosphorylated happen in a cell as a result of DNA damage, right? So no, remember RB being phosphorylated is going to release E2F, which is going to allow us to go through the cell cycle. So if we have DNA damage, we do not want to do that. All right, so chromosome condensation, oh, I can't even pronounce, occurs at the beginning of this phase, right? I kind of showed you the answer here, right? But there was M phase. I think that's just like a fact to know, make sure you know, right? C here, I keep on showing you the answer. C, CDK activity is at its lowest level at the entry point of this phase, right? So this would be G1, right? Because remember, we need active CDK throughout the entire cell cycle, so we expect CDK activity to be the lowest during G1. So which of the following statements correctly describes P53, right? So I'm going to pause here for a second so you can read them because they're a little bit longer. But this was I think this was a final exam multiple choice question. So maybe you'll get lucky enough to get it again this year. And if you read that, you would know that D, activation of P53 leads to inhibition of cyclin CDK complex, would be the correct answer. Any questions on A through D? And just a reminder, next week, we're going to be covering S, G2, and M. So that's all I had for the prepared slides. I'm just going to stop recording.